Sorry. Awesome. Okay, even better. Good morning, everyone. Welcome to this last session of this pre-GRC Quantum Biology lectures. Thank you again so much for this faithful group of co-organizers, Susanna, Nathan, Ale, have been already, who have been here every single day, Arat, Abazout, Irina, Omid, who have also helped a lot. So thank you all for making this. So I am just going to say that all the slides and the link to the lectures will be posted in our school website. So just please be patient as we collect the slides and put the videos online. But it's been a great pleasure talking to you these last days. So without further ado, let's introduce the first speaker today, Vahid, who's going to talk about so today also it's really early. I'm tired today, sorry. Today the theme is like Quo Vadis quantum biology, is like looking forward, right? So we're going to talk about some relatively new modalities of quantum biology, and we hope to start hearing more about theory and experiments for those modalities very soon. So first of all, we're going to hear about biophotons, then about something funky that has to do with spins.
Clarissa, I'm sorry, I'm the next speaker, I think, because yeah, I explained the problem to you before.
Yes.
So yeah, in 1 hour, yeah. So I'm not the first speaker, I'm the next speaker. Yeah, the first speaker is not a person, if you check it again.
Oh. Oh my God. And you don't go right now.
I explained the problem to you before, that in this time, maybe I should leave the meeting for half an hour and then I will join again.
Oh, you have another engagement.
Should be Dave.
Oh, no, that's fine. That's fine. David Waldeck should have been here at 7 a.m. Oh,
yeah, maybe
okay.
you should just wait for him.
Waldeck, are you joining the Zoom room for the GRC 7 a.m.? No.
Well, in the meantime, how has everyone enjoyed the event so far? I see one heart. Brilliant. Thumbs up. As we come to the close of the week, is there anything anything else you'd like to see? Anything you thought was particularly well done or that you would like to see done differently if we run this event or a similar event in the future? Don't be shy.
We take constructive criticism.
No, we take it very well. We love constructive criticism. Maybe it would be a good time to release the feedback form. If you have comments and you would prefer to share them confidentially, we have a feedback form that we would love if you could take our survey. Susanna, do you think you could help put that out? Thank you.
Yeah, I'm just finding the link for that.
Great. Okay, we have a nice comment about the group discussions. No, I'm glad those were appreciated. Emma expresses her bias. She absolutely loved it. Great to hear.
Right, I'm going to paste a link to the feedback in the chat.
Wonderful. I'm opening it right now. Oh, excellent. Okay, well, while we wait for our first scheduled speaker, if you don't mind, if you have some time and you're happy to fill out the online summer school feedback form, we would love to get your comments and suggestions. Also a little bit about your background, so we have some sense of what areas of research we've attracted to this event. So thank you very much in advance for that. Again, we really appreciate any comments or criticisms you may have. Oh, excellent. Someone has recommended hands-on experience through labs. Of course, if we had an in-person summer school at a location, we could do lab tours. That would be great. And general comment from Austrians. What a great summer school. Thank you. No, that's wonderful to hear.
Folks, the first speaker has been contacted by email. Okay, so maybe in order, while we wait, and the first talk is about this phenomenon that is called chiral-induced spin selectivity. So since our lab is interested in this phenomena, and Ale, I'm looking at you. Why don't you give us while we wait for David Waldeck, who's one of the main people in this field, why don't you start explaining, like, in very, very short terms, like, I know it's early here in California, but can you explain in simple terms what this chiral-induced spin selectivity is about and why it's surprising?
Sure. Put on the spot right now. Just woke up. Thank you for that, Liz. But no, yeah. So I'm sure they will be like 1,000 times better in explaining that. But yeah, this chiral-induced spin selectivity is an effect that was discovered in DNA in 2012, I believe. And it consists basically in the fact that if you put an electric current through a chiral molecule, or like a helical molecule, usually, like DNA, for instance, you get that at the end of the molecule, when the current is passed through, you observe a spin polarization, which means that the current that goes in has 50% spin up, 50% spin down, goes through the molecule, and maybe at the end of the molecule, it will have, I don't know, 90% spin up and 10% spin down. And this is related to the handedness of the molecule. So if the molecule is right-handed, it will have the spin polarization, I don't know, up. And if it's left-handed, it will be this. I will have the spin polarization pointing down. And so the amazing thing of this phenomenon, the charming thing, I would say, is that we have no theory right now that can explain such a large magnitude of the spin polarization. So usually, at least from my experience in the quantum field, you have like these nice theories that say, oh, you have this amazing effect, which is gonna change the world because you can have this great signal. And then you go and measure it experimentally, and it's like 3-4 orders of magnitude lower. And this is exactly the opposite. So we have the theory that underestimates the effect. And so I think it's a very exciting phenomenon which doesn't get enough attention. And yeah, so we need to find ways to really figure out what's going on. Is it like thermally assisted? Is the spin locked in to the chirality of the molecule in some way to give rise to this huge spin polarization? And yeah, so that's pretty much the gist of it. Am I missing something, Clarice? Okay. But yeah.
I'm starting to get a little bit worried that our speaker is not here. Vahid, how much time do you have before you have to leave and come back?
Maybe I will leave in 5 minutes, then I will be back at after half an hour. I'm so sorry.
I am trying to find things that we can do before. So, okay, while we continue to wait for David for the beautiful lecture, he gave a similar lecture in our weekly seminars on Quantum Bio. So another advertisement here. If you want to join the Quantum Bio community, if you want to join the Quantum Bio community, you are free to subscribe to this Google group. Oops. You're free to subscribe to this Google group where we talk about our weekly seminars and so forth. And for people who have just joined, we are waiting for David Waldeck, our speaker for chiral-induced spin selectivity. After learning about chiral-induced spin selectivity and about biophotons, we have a slot to talk about teaching and learning quantum biology. So maybe while I keep an eye on my email to see if they've sent something, I would like to propose that while we wait, we could start having this discussion about what it means to learn an interdisciplinary field, right? What does it mean to start working on an interdisciplinary field? And maybe we could have the discussion as an entire group here. Yeah, you can unmute yourselves and stuff like this. So, and again, this is why we're waiting for David. John, in the last slot of today, we're going to have John Joe McFadden, who's one of the authors of the book Life on the Edge. Life on the Edge, which is a very cool book that is almost 10 years now. And for everyone who's interested in quantum bio, it's a very nice layman's book that talks about the different flavors of quantum bio, no math or quantum mechanics necessary, right? And if you have been here in this school all the days, right now you know a lot of what's already explained in this book and in a lot of mathematical details. So John Joe is going to be here because he's also the director of the first ever quantum biology doctoral training center in the world. So the University of Surrey in the UK, I think about 3 or 4 years ago, they started a quantum biology doctoral training center funded by a big foundation in the UK. This meant that they had, I think, doctoral positions officially in quantum biology for about 10 to 15 people. So to the best of everyone's knowledge, this is the first time that a cohort of people working in quantum biology were hired at the same time. So one of the things that he's going to talk to us about is really how this cohort is advancing, right? What it means to make to work on an interdisciplinary science. And I know that their cohort has people from many backgrounds. So I am also going to put Raishin on the spot, who just joined. So Raishin, good morning. So we have a small problem here because our speaker for the time hasn't joined in yet. So while we're waiting for them, they have been duly contacted. We thought we could spend some time talking about what it means to learn an interdisciplinary field. And since John Joe, the director of the Quantum Biology Doctoral Training Center, is going to talk to us later, I was wondering if you, as one of the students in this very first cohort, could share with us your experiences and how did you enter this field? What's your training been like? How have you been, in such an interdisciplinary field, everyone is learning something at the same time, either the physics or the biology or the chemistry. So how have you managed to learn the parts that you weren't necessarily trained on? So can you tell us a little bit about that while we wait for our speaker?
Sure. So I came at the second year of the doctoral training center, and my background is in physics, but my course ended up being much more chemistry. So I suppose if it wasn't for the pandemic, I probably would have been able to do more for physics and mathematics inside my project, but as it was, I ended up kind of going down the more the chemistry route, and the challenges there were definitely like learning the codes. So I feel every time I meet a chemist, I'm always talking about what codes they use or how to implement their methods, and every time it's the physicist, it's always about what equations you're using, and then when it's biologists, it's kind of asking them so many basic questions like why am I reading so many papers where they're using E. coli as a cell? It's kind of broad questions to help me understand how my research fits into the science. There's a lot of revenues that I've learned stuff like these training courses have been great for telling me the theory and the broad scope, and then separate training courses for learning programming. It's a really wide range.
So during your training, you didn't have formal graded classes.
So I went to some of my supervisor's lectures, but no, there's no strict training course. And of course, in first year of the pandemic, it took quite a few months before alternative training courses could be set up online. The first year is really difficult, but the training courses online really helped me get them. We're back in track. Start. But yeah, there's positives of the pandemic too. Like I'm not sure, I would have been able to do conferences like these, or anyone would have put them together if it wasn't for the pandemic. It really it changed academia.
Yeah, it's all online. Incredible, eh?
To travel at all. Right, yeah, like I'm not able to go to Texas next week just because there wasn't enough funding, so I'm really grateful to have this instead. It feels like I've almost got to the experience.
And you're hosting another conference on quantum biology at Surrey later this year, I think, aren't you?
Yeah.
Are there details available about that could be shared with people here?
Oh, sure. So because of my funding, I'm not sure if I personally will actually be around for that. I might be out of Surrey, though I'll try to still attend. I can give me a few moments. I'll share a link in the chat maybe.
That'd be great. Are there other people here who have done sort of specifically quantum bio PhDs or courses in their undergraduate? I know we've all come from quite a mix of fields.
I was fortunate enough to win a nanotechnology scholarship that allowed me to transition my original plan for my PhD, which was mainly in quantum information, to joint work in quantum information processing and biological electron transfer, looking specifically at the way the kind of modeling and analysis framework could be applied to something like an electron transfer reaction, where again you see some of the same features, most notably the need for energy levels to be the same during an electron transfer reaction, similar to the qubit states in a quantum computer have to be degenerate in order to avoid decoherence or losing the phase relationship between those states. So it was very interesting and a great opportunity for a PhD.
Hello, everybody. Hi, I'm Zoya. I was wondering if anybody is teaching for undergraduates any classes like introductory, and it'd be nice to exchange. We're trying here, Michelle is here also, I see in the meeting, but we have a group at University of Waterloo in Canada, and we're trying to introduce this a little bit of introduction to undergraduate fourth year. Is anybody teaching anything at all? Would be nice to exchange expertise. And the books that Clarice, you mentioned, it's a little bit for physics students, maybe it reads like a novel, more, not like a physics course, but is there anything, can you guys share if anybody is teaching anything at all?
So did you manage to make a course called Quantum Biology?
Yes, well, Michelle and I, we have this first trial course for fourth year students, and just collectively, but mostly around quantum neuroscience and a little bit of introduction to other areas. But we are planning to develop it as a full course, and I just thought it would be nice to see if anybody is developing anything or is there any material. This is nice lecture course that you, Clarice, put in together with your team. This is really nice for graduate students and good discussion forum as well. Another thing I thought would be nice to have I don't know, maybe you have it already, like a database, people with different expertise. If, for example, students are searching for some questions on ion channels or spin selectivity and things like that, and at least for some main players, and it would be interesting to have this as well.
I'm curious, do you have, what do you use as a textbook or course notes, selected papers for the class? Zoya?
Well, Michelle and I, we just use the books that are available, and we just selected material. It's only lecture notes. It's not really a full book. That's why I'm asking it was, but several books on life on the age, one of them. But also for some specific things, you have to go to the paper and then present a little bit of extract from the papers. And I don't think there is a suitable book for teaching, but it would be nice to teach fourth year undergraduates in physics to get them in, and they are interested. They actually like to learn. As graduate students, they already expert in their field, and they come and they can, but in introductory course would be nice. What about you, Nathan? Do you teach anything at home?
I have only done some substitute teaching. I haven't taught a whole course yet. I look forward to the opportunity. I've certainly thought a lot even for this
I heard
event.
your lecture, it's really nice. So I thought it would be, well, maybe some of these lectures, at least material we can take and adapt, and I don't know.
That sounds great. I know I'll see Michelle at the Quantum Bio meeting next week. I don't know if you'll be
Oh,
there.
he's here. I think he has a meeting to go, but maybe he already gone.
Wait, can I recommend someone from the university? Dr. Yang Chan. I think he's one of the organisers of the Texas conference, and he's definitely been organising a quantum biology course to teach at the university. That's all I'm saying.
So
Yeah,
I'm so sorry to cut this discussion. We will go back to it. So our speaker for chiral-induced spin selectivity, we couldn't reach him, so I'm so sorry. So we have a makeup speaker that we've just organized, Alessandro. So here's the background, and I'm going to put those lecture not about chiral-induced spin selectivity. But last semester, our group organized a series of lectures going through the chapters of this book, Life on the Edge, and Alessandro presented the lecture about one thing that we haven't talked about a lot about in this school, which is, well, we did talk about that. So we talked about noise-assisted tunneling. And one of the things that is thought to be regulated by noise-assisted tunneling, it's not super proven, but there's correlative data that points to that, is olfaction. So Ale has a presentation about olfaction, and I sort of volunteered him to present this lecture. So thank you very much, Ale. We are going to hear about olfaction maybe for the next half hour. Then we're going back to biophotons, and then we can have a whole hour of discussion of quantum biology, how to teach, how to learn. So thanks for that. Sorry for the thing about the chiral-induced spin selectivity. We'll try to provide you with a lecture on that. And Ale, take it away.
Okay, yeah, this is definitely unexpected, so please bear with me. I'll do my best. Okay. So this was not in the plans and so on. So I'm just recycling an old presentation that I made for this.
Sorry, sorry, Nathan, how did you call it? Faith-based management.
Yes, that's right. Alessandro, this is your big break.
Amazing. Yeah, so we had this nice seminar series, sort of similar to this, but based on the book Life on the Edge that Clarice mentioned before. And this is chapter 5 of that book, a brief summary, which is called Finding Nemo's Home. And there were spoilers, so I had these all quiz, sort of. And so the whole picture starts with Nemo, right? I hope everybody knows this guy. And I don't even remember what I talked about. So, okay, right. So this is the thing. You have a male and a female. This is the reproduction of how are these fish called again? I don't remember.
Poundfish?
Anemone fish, something like that. Yeah. So you have a male and a female. They love each other and everything, and so they make the female lays the eggs, and then her job is done. She just goes away, and then the male comes and he fertilizes the eggs, and then he sticks around for a while to protect the eggs that will become the babies. So then you have the formation of these little embryos, whatever, and then once they hatch, they're actually scattered all over the coral reef. And so maybe at some point they find each other like all over the place. And the thing is that when they're older, when they grow up and fully develop into the fish, they always go back to their original coral reef. So what do you think they use for orienting themselves and go back to the same point? Like they're all scattered here where the stars are, even like hundreds of miles away, but they all go back to here where they're born. So what do you think they use? And maybe Google Maps like we do. Maybe they like to ask directions, and the fish in the sea are like the people in LA. They like, for reference, people in LA really love to talk about streets and directions. I don't know why. Maybe they orient themselves with the position of the sun or the moon. Maybe with ocean currents. Maybe they use just visual clues. Maybe they smell their way. Maybe they use sound, or they use the Earth's magnetic field. What do you think they do? Lots of options. Okay. Okay, yeah, I think it was a bit, there was a bit of a spoiler about smelling and odors. So yeah, the answer is actually smell. So I will briefly talk about in these few minutes that I have about, yeah, the general role of olfaction in a broad way, and the history and the development of the olfaction theory. And while I do that, I will introduce a little bit of quantum tunneling. I will go very fast because we had pretty much a whole day yesterday talking about quantum tunneling. So yeah, I had a whole session of introducing myself because I didn't have enough material to keep an hour occupied, but yeah, so I can very briefly go through it. I'm originally from Italy, and I started off as a mechanical engineer, which is very weird. But then I thought, you know what, I really don't like money, so I'm gonna go into physics. So I did a master's in engineering physics, and then transitioned to a PhD in condensed matter physics, studying mostly spintronics and two-dimensional materials. Then I moved to something very weird, which is topological quantum computing, where we try to build qubits that use the teleportation of these weird Majorana particles, are called, to implement qubits that are robust enough. Didn't work, by the way. But then I found out of Clarice's work and quantum biology. I fell in love, and here I am. Okay. So yeah, the common denominator of all my research is most of the techniques are based on quantum tunneling. And so I will try to convince you in this lecture that the olfaction mechanism is somewhat related to quantum tunneling. And the bio part, I will probably get wrong and not very strict. So please forgive me for that. But maybe the quantum tunneling part, I can give you some insight. So, first of all, let's start with the importance of olfaction. Olfaction is a very, very important thing. And I don't know if any of you had COVID, I'm guessing you had at this point. But the first time I had COVID, I lost my sense of smell. And in that moment, I realized how important it is and like how much we give it for granted. So because you smell to enjoy your foods and drinks, and so I realized that everything tastes the same without smell. I could taste like salty and sweet and so on, but literally like eating pizza or broccoli was the same thing for me when I lost the sense of smell. So I never ate so healthy, but yeah. But it's not the only thing that we use olfaction for, and it's also a defense mechanism. So if we smell something bad, it means that we probably shouldn't eat that. Like we should stay away from that from an evolutionary standpoint. Makes sense. So if I smell something bad, it means no, that thing shouldn't be in my mouth. Like for instance, pineapple pizza. That's illegal as an Italian. But for other species, we're pretty bad at smelling things actually as humans. But for other species, they use it as a survival mechanism, actually. And sharks, for instance, have, I don't remember the exact number, but something like 80% of their brain dedicated to olfaction alone. And they're super good at smelling a drop of blood even miles away. And also, animals can actually smell pheromones. And so they know when a female or male or whatever are ready for mating and the survival of the species. So after this introduction, I want to go back to Nemo. So we have that Nemo finds himself in a random place in the ocean and then needs to go back to his coral reef. So, and we already told that it's based on olfaction, but how do we prove that? So what researchers did was to do something like this. You have a sort of a stream with two branches and the flow that goes towards Nemo. And they put two very similar reefs. One is the one found in the native coral reef, and one is just another. And then they saw that the anemone fish always swim towards the native reef. So this is a lab setup. So they have no visual clues or sounds or whatever that they can use to actually find the native reef. And they don't really have a magnetic compass, and in the lab setup, it doesn't really make sense they could use that. And so the ocean currents, they're not there in the lab setup. So the only thing that is left is actually the smell. So they also did that in a different setup with salmon. And so researchers did that in rivers that have branches, and turns out that also the salmon always wants to go back to where they were born. I don't think, I think. Well, they want to go back to their original place. So what they did is they went fishing for salmon, and then they tagged them and release them here at the bottom of the branch. So they always found that they were going back to where they were captured. And so they repeated the experiment, but they blocked their nostrils with some cotton swabs. And they saw that actually they had no preference of going one branch or the other anymore. So this is further proof that fish use olfaction as their navigation tool. So, but how does olfaction work? And let's go back into humans. We mostly care about humans, right? And so here we have that the first step is that we use our nose to suck in the air. And then you have that the nose actually doesn't have an active role in the olfaction system. It's just there to filter the air a little bit, but it doesn't actually provide the olfaction mechanism. And so if you've ever wondered about that, yes, also Voldemort can smell. So everything that is relevant happens in the olfactory epithelium. And this is structured like this. And you have odorant molecules that go in and they get captured by the odorant receptors. So once this happens, the ion channel of the neurons open, and then they give the neuron fires and gives a signal to the olfactory bulb that then processes the signal and sends it to higher parts of the brain. So what we want to focus on is the part where we detect the odorant molecule. I would say this is the most fundamental part for olfaction. So the first question that we can ask is, can a single receptor identify every odor? Or do we have like specific receptors for a specific odor? So we know that humans have roughly 300 different olfactory receptors, while for instance, rats have about 1,000. And this is part of the Nobel Prize work from Axel and Buck. And so each receptor is actually a little different. So maybe this is a hint that we can use them for single odor detection. But this doesn't really add up because we can detect roughly 10,000 different smells. So how can we do that with just 300 receptors? So what they did is they investigated genetically altered mice where you have that an olfactory neuron will dye blue when it's activated. So they can put a smell and then they just see which of the neurons turn blue to see which one was active in the process. So for a single odor, you have that only 1 in 1,000 receptors, that they have 1,000 species, is dyed blue. So turns out that our olfactory receptors are specialists. So an olfactory receptor can detect one single smell, right? So to answer these, think of 10,000 different smells. We have that actually they found that odorants can activate multiple neurons and that single neurons can detect several different odorants. So you have that these 10,000 different smells are probably arising from different combinations of the activation of the different receptors. And this is how I picture it. So let's say that this neuron detects yellow, this one detects blue, and so what your brain receives is green. So the mechanism that was first proposed and is one of the leading mechanisms throughout history is the lock and key mechanism. So the idea is exactly like a lock and a key. So you have your lock, which is the receptor, you have the molecule, which is your key, and only the right key goes into the receptor and can activate it or open it, right? So with a molecule, it's going to be something like this, where you have that the shape of the molecule is going to be the inverse of the shape of the receptor, so that it can actually go there, lock, and this activates the detection mechanism. So there are few limitations for this, which are that you also have that completely different molecules, like these ones, for instance, have exactly the same smell. For instance, these all smell like musk. And on the other hand, you have that very similar molecules have completely different smells. These ones are almost the same, just this group is missing here, and this one smells like urine, and this one is odorless. So the lock and key mechanism cannot really explain this. So then this guy comes along, and Malcolm Dyson, and he investigated this group of mercaptans, they're called, and these all have in common that they have a terminal sulfur hydrogen bond, and even if the molecules are very different, they all have this terminal group, and they all smell like rotten eggs. So they all smell the same. So maybe he proposed that the receptors actually pick up not the shape of the molecule, but the vibration of the molecular bonds. So, but then when in the late 1920s, there was no way of picking up vibrations of molecules. So we have no way to measure that until Raman spectroscopy was invented. And you have that the idea behind Raman spectroscopy is that you shoot a laser, monochromatic laser, single energy on a molecule, and you have that most of the light will be scattered elastically, so with the same energy, but you have a small portion that is scattered inelastically and can lose or gain energy based on the interaction with the molecule. And this part is what you collect. And if you collect the light intensity as a function of the wavelength, so the energy, you have something like this, where you obtain a spectrum that identifies your molecule, because each one of these peaks corresponds to a molecular vibration. So now we go back to the idea of our olfactory receptors picking up molecular vibrations. And we see that here we had this sulfur hydrogen bond, and actually it seems like all these molecules share the same Raman frequency corresponding to the sulfur hydrogen bond here. So maybe the olfaction actually works like a Raman spectrometer, right? And sure, but there is a but, which is humans don't really shoot monochromatic laser beams from the nose. So the idea is cool. We have the vibration that is somehow related to our sense of smell, right? But the Raman spectroscopy cannot really explain that. So this is where quantum tunneling comes into play. And there were much better descriptions that I could ever make yesterday. So I'm just gonna go through these very quickly. But I always picture quantum tunneling as throwing a ball on a wall. So if I throw a baseball on against the wall, I can throw that like as many times as I want, but this ball is always gonna bounce back. This is because I have the wall, which is an energy potential, so an energy barrier. And I can picture this exactly as a potential energy barrier, where the potential energy is the gravitational potential energy. So the only way I can throw this ball on the other side of the wall is by throwing it up above the wall, right? When we shrink this down to the nanoscale and think about electrons and these being less than a nanometer or so, we actually have that there is a finite probability of the electron just going through on the other side. Sorry. So it's difficult to see when we think about particles, but as was mentioned several times before this week, electrons are not really particles. They're also waves, right? And so we can think of this as a probability distribution, like this yellow thing. And if we shoot it against the barrier, we have that there is a non-zero probability of finding these here on the other side. And so most of the time, these will bounce back. But some of the times, you will have this probability of just the electron going through. And this is roughly how I picture tunneling. It's not like a very strict picture, but it can give you an idea. So for the math aficionados, this is the tunneling current equation. And the thing is that you have a lot of dependencies, like the density of states of your sample, of the different materials. And you have this distance, the energy, and whatever. And if you engineer well enough an experiment, you can study each one of these dependencies. And so this is some of the applications of quantum tunneling in physics that I am most familiar with. And you can get nice atomic resolution images with these scanning tunneling microscopes, they're called. You can actually manipulate single atoms and put them in the way you want. Like this was done at IBM. They even made at IBM Almaden here near San Francisco the smallest movie ever that you can find on YouTube. And these are single atoms that they just moved around and made a stop motion picture. But you can also do magnetic resonance and spin manipulation with single electron resolution, which is what we're trying to do at UCLA right now in my project. And we want to do this to study the currently induced spin selectivity, which you should have known something about right now, but speaker went AWOL, so not gonna talk too much about that. And yeah, so but apart from these parentheses, just go back to the basics of quantum tunneling. And Nathan explained very well that we have that the quantum tunneling is an elastic process. So you have this donor and acceptor, right? And you have an electron here, and these can tunnel through the barrier. But this tunneling needs to be at the same energy. So if I have another energy state here, this tunneling process cannot occur because it violates the conservation of energy. So, sorry. What about I put something in between? So if I have a molecule in between that has a particular vibrational state that matches the difference between the energy in the donor and the acceptor, then I will open up a tunneling path, because then this becomes acceptable because the energy is conserved. So this idea was proposed in the context of olfaction by Luca Turin. And this is sort of the mechanism. You have the donor site in one part of the receptor and the acceptor site in another part. So let's take this molecule, and let's say that this energy difference matches one of the vibrational states of this molecule, which is, let's say, 76 terahertz, right? So what happens is that because of this vibration, the tunneling pathway is opened, right? Where the vibrational energy is the energy of the donor minus the energy of the acceptor. So what he proposed is he found another molecule which is completely different, which is the camurine, both in shape and in chemical profile, but has a very similar frequency, vibrational frequency. And what he did is, what he proposed is that these molecules, since they have sort of the same vibrational mode, they will smell the same. And it turns out that they actually do smell the same. And they all smell like rotten eggs. This has a sulfur hydrogen bond, is one of the ones that we saw before. And so this is amazing evidence that vibration and tunneling could be related to how we smell things. And this is amazing, but it's still not enough to convince all the skeptical physicists and biologists and chemists out there. So what he proposed is to study the isotope effect. Am I running late? Maybe yes. So very quickly, we can replace in molecules hydrogens with deuterium, which is a heavier isotope of hydrogen. So it's where the hydrogen nucleus has a neutron on top of the proton. And this is twice as heavy as the hydrogen. So these two molecules, for instance, will have identical shape, identical chemistry, but they will have different vibrational modes because of the mass of the deuterium. So his idea was, if we have different vibrational modes, but the same molecule, these compounds should smell different. And so what he did was take this cetophenone molecule in the normal form, undeuteurated form, and in the deuterated form, where all the hydrogens were replaced by deuterium, and he just smelled it. And he was amazed because he could tell the difference between the two in smell. And so he was very hyped. Well, yes, my vibrational theory is right. But of course, this is not enough to convince other scientists, because you can be biased, and it's just your perception. So let's just have others smell the difference between the two. And turns out that other people could not really tell the difference between the two compounds. So this is sort of a problem. But still, I wouldn't consider this much of a scientific evidence, just people smelling things and reporting. So he was like, okay, let's make things actually scientific. And he used Drosophila, so common fruit fly, for this experiment. And so the first question is, can the flies actually smell this ACP molecule? So in a very similar manner as the two branches for the fish to swim into, they had a tube with two solvents, with two odorants that are that flow towards the center. And so they put the flies here, and they either go one way or the other. And it turns out that here you just have the solvent where they put the ACP, and here you actually have the ACP, and they flow them, and they see that the flies tend to go towards the ACP. So they actually like the smell of this, and so they fly towards there. So what if I do this with deuterated compounds? Does something change? Can they tell the difference? And this is with 3 deuterated ions, with 5, and with all 8 hydrogens replaced by deuterium. And you can see that the distribution of the flies inside the tube is very different in the different cases. So can they distinguish them? You can already tell that they can, but they actually put it to a direct comparison where you have the normal ACP and the deuterated ACP here. And you can see from this graph that there is a very strong preference of flies going towards the ACP. So yes, olfaction seems like it's linked to the vibrational properties of the molecule. And but there are still some big limitations to this theory. And one of the biggest limitations is related to chirality. So these two molecules have the same molecular bonds and the same vibrational properties. The only thing that changes is the direction of this chemical group, which is in one sticking out of your screen and in the other going into your screen. So these are called enantiomers. So they are the same molecule with a different chirality. And actually, these molecules, for the theory of vibrational theory of olfaction, should smell exactly the same, but they smell different. This one smells like lemon, these more like pine cones. And so vibrations alone turns out that they cannot explain olfaction. So the shape of the molecule must also play a role in the detection. And so what I wanted to tell you today, even if it wasn't planned, is that we have a very strong evidence that olfaction is related to the vibration, both the vibration and the shape of the molecules that we need to detect. And we have that the most widely accepted model right now seems to be a combination of the vibrational and the shape model, where only parts of the shape of the molecule is involved in the detection of the odorant. And we still lack direct experimental verification that quantum tunneling is occurring in the detection of the of the molecules. So there's one other big limitation is that the structure of the olfactory receptor is still very much unknown. And but it's amazing to see that every life phenomena that we give for granted, like olfaction, can be regulated by weird quantum effects such as electron tunneling. And yeah, I had also these sort of motivational things for the students in the course when I presented, which was that if you're an undergrad or like if you want to get involved in research, I think quantum biology is a great field because it has a lot of unanswered questions. And unanswered questions are what you want when you do research, right? And so I think in research, if you want to do it, the thing that you should look for is a question that you find so interesting that you're willing to spend your life trying to answer it. So hopefully with this introduction and everything this week, you found a question that you're interested in and you would like to look at in the future. And thank you. I will finish here.
Can we give a big round of applause to Alessandro, who rose to the occasion of a missing speaker and improvised? Thank you so much, Ale.
No problem. Yeah, I didn't really remember everything I was saying, but hopefully it wasn't too bad. Yeah.
Thank you, Ale. So I think we're ready for we still owe you a lecture on the chiral-induced spin selectivity. So let's see what we do about that. Thank you, Ale, so much. So we are ready for our second speaker, Vahid, and you're in for a treat because we're going to talk about biophotons.
Yeah, hi everyone. Thank you so much, Clarice, for your introduction. And yeah, I finally could join the meeting. Let me first share my screen. Okay, can you see my slides?
Yes, but not in presentation mode.
And now? Is it okay?
Yes, yes.
Okay. Just have to displace this taskbar. Okay. It's a real pleasure being in this exciting event in this pre-GRC school, and it's really sad that I missed the meeting in person next week because of the US visa issues. But I'm happy that in this school I can speak. First of all, let me thank the organizers, especially Clarice, Yung Chang, and other organizers, and especially trainees like Abbasat, Omi, Nathan, Bethany, perhaps, and other trainees. I know that it needs hard work and efforts to follow up all issues. Okay, my name is Vahid Salari. I'm research associate at Institute for Quantum Science and Technology at University of Calgary, Alberta, Canada. Today, I will talk about biological ultraviolet photon emission. But before that, let me give you a short introduction about our group. I don't know why it doesn't. Okay. Yeah, I'm working in the quantum biology lab, which is a part of a bigger lab, which is called Quantum Cloud Lab, which is an experimental lab working on quantum technologies at the University of Calgary. On the other side, also we have a cluster of collaborations, mainly working with Professor Christoph Simon, who is a theoretician, in the Quantum Cloud Lab, which is leading by Professor Daniel Oblak, who is experimentalist. And this quantum biology group, the website is quantumbiology.ca. This is actually a new group. Just a few months ago, we created this group, and yeah, there are some activities that are still ongoing, and you will see new results soon. There are different funding agencies that they support our work, especially regarding this ultraviolet photon emission. We could get funding around $2 million so far. And hopefully, because of this national quantum strategy in Canada, that they approved $360 million on quantum technologies, hopefully maybe we can get some of the fundings on other aspects of quantum biology. Also, there's a quantum city in the campus of University of Calgary. This is an environment to commercialize some quantum technologies, and one section is on biomedical issues and biological sciences, and perhaps they will fund also quantum biology topics. In that case, the government of Alberta invested around $23 million, and the other side, the company called Emphasis, they will support this quantum city up to $70 million. So we will have money, and it's good news for quantum biology in Canada, especially at the University of Calgary. And let me give you another introduction about the projects we're doing at Quantum Cloud Lab. The Quantum Cloud Lab mainly is working on quantum communication and quantum memory, on quantum key distribution, quantum cryptography, single source, single photon sources, single photon detectors. And a part of that is Quantum Biology Lab. Or maybe this is a question for you, that why Quantum Biology Lab exists in this Quantum Cloud Lab. The main reason is that because the technologies used here for single photon detection are so sensitive that they can hunt single photons. And in that case, we can use it also for detection of photons emitted from living systems. Okay, maybe another question you will have, that do biological systems emit light? The answer is yes, but we have different kinds of light emission from biological systems. It can be induced or it can be spontaneous. In induced state, it needs external excitation to cause photon emission from living systems. This external excitation can be light, can be laser light, can be chemical factor, chemical agent. It can be electromagnetic field, it can be ultrasound. So it can be even stress or injury. In spontaneous emission, it doesn't need external excitation. It's emitted spontaneously, and the mechanisms can be different. The famous one is black body radiation or thermal radiation, that you can see this kind of light by infrared camera and not by your naked eye. And regarding the induced light emission, also the intensity of this light is not so strong. Again, you need some specific photodetectors with high quantum efficiency to see this kind of light, but maybe not so high sensitive. And the next one in the spontaneous emission is called bioluminescence. It has the highest intensity among them, and it only can be seen from some particular species. For example, like firefly, like jellyfish, like phytoplanktons, and some other kinds of plants, that they can emit light because of some specific reactions, like enzymatic reactions, like luciferin-luciferase reactions, or non-enzymatic reactions like photoprotein and other mechanisms. This kind of light can be seen by naked eye. You don't need some specific detectors to see. The last one is called ultraviolet photon emission. This is the topic that I want to talk about it today. It has the weakest intensity, and the mechanism is different. It's mainly because of reactive oxygen species and also free radicals, triple carbonyls, dimolar singlet oxygens, singlet excited pigments, and some other unknown mechanisms. And still the research in this context is continued. But to see this kind of, or just to detect this kind of photons, you need high quantum efficiency photodetectors. There are different kinds of instruments that can help us, like PMT, like EMCCD, CCD, and also avalanche photodiodes. and some of the photodetectors that like what we are using in our lab, like superconducting nanowire single photon detectors or SNSPDs. So I will talk about it later in the next slide. Maybe in different publications, you see different names for these kinds of emission. Because people don't know maybe the mechanism, and sometimes even they call it bioluminescence, which is not correct. Or spontaneous chemiluminescence, or biophoton, biochemiluminescence, self-bioluminescent emission, or photoluminescence. There are many names in this context. So the community a decade ago decided to choose a single name for this kind of emission, and they called it ultra-weak photon emission, or with abbreviation, it is UPE. So I'm talking now about UPE. You have a feeling about the intensity of light. Let me get back again to bioluminescence that I told you that even without any instrument, you can see it. On the left side is an image taken by a satellite, one part of the Earth and the Indian Ocean. Okay, on some parts you can see some lights. These are the city lights. Okay, I'm not talking about that. But if you look at the sea in the night, there's a shiny part, and the length of that is around 250 kilometers. And this light can be seen even from the satellite. These are actually because of phytoplanktons and marine entities, that the intensity of light is so strong, and with the so vast area on the ocean can be seen. And also, these are some examples that I talked about it, like jellyfish or fireflies. And regarding the infrared picture, just one important point here, that maybe sometimes people say that how can you make sure that this kind of emission is not thermal radiation? If you calculate the blackbody radiation in room temperature, the first photons are emitted just above 1000 nanometers. So the main spectral range of detected UP is mostly detected in the range 200 nanometers to 800 nanometers. But this doesn't mean that there's no biophoton just on bigger wavelengths, for example, 1000 and 1500 nanometers. In that case, just because the overlap of thermal radiation and also the detection efficiency of the photodetectors here mostly don't help us to detect this kind of photons. So this is an unknown place, but fortunately in our lab, we have something new that it can detect these biophotons in this range, in the range of near infrared. So hopefully we get some results, that we have some preliminary results on tadpole brain via SPDs, because it uses some kind of cryogenic environment that it can diminish the amount of dark current or thermal energy to 4 Kelvin or even at millikelvin. And then the amount of dark current will be diminished, and the contribution for thermal energy will be diminished. So you will see the signal to noise ratio higher, and you can detect biophotons. Okay, regarding the light emission from plants, here is an example. On top, you will see delay luminescence. This is actually an image on the website of Hamamatsu, which creates the photo detectors and the field of PMT and also CCD sensors is famous. On the left side, if you look at that, some part is yellow and the rest is green. So the yellow part is actually because the chlorophylls of the leaves are removed. And then it is illuminated by sunlight after a few seconds. And then it will be imaged for a few minutes. And you will see such image under the EMCCD. You will see that on the parts that there's no chlorophylls, there's no light emission, almost no light emission. It shows that the main source of delay luminescence is from chlorophylls, and it can take nanoseconds to a few minutes in this case. And the intensity of delay luminescence can be 1 or 2 orders of magnitude stronger than UPE. And on the lower image, you see in the first image is the leaf under room light. The middle one is the delay luminescence, which is a stronger, and then the last one, which is called UPE. Actually, the first one is after 1 minute being in darkness, and after a few minutes, let's say 5 minutes, this image is taken. And then after 27 minutes being in darkness, for 20 minutes exposure, this image is taken. So this shows that the mechanisms are different and they're not similar. And the intensity of photon here is only few photons to 10 to 3 photons per centimeter square per second, which is the lowest. And it can, but this UP can take several hours and even days, and maybe always exist this kind of photons. So people, even for a few days, they recorded these biophotons, and it was spontaneous and still continue. It's a long-lasting kind of emission. Regarding UP by plants, let's say light emission by plants, this is a diagram, is intensity in terms of time. There are different states for emission, and there's some cottage just to show that there are different mechanisms. The first phase is prompt fluorescence, and it takes only in the order of nanoseconds, and quickly it drops. Then delay luminescence started, and it takes between 5 minutes to 15 minutes. The intensity is still high. And then after 15 minutes, the intensity reduced to a constant. Then for the duration of 1 to 5 hours, this emission is almost constant. This is mainly because of chlororespiration. And then after five hours, then some oscillatory behavior is started, and which is actually a long-term UPE, and it is mainly because of free radicals and lipid peroxidation in mitochondria, the most contribution. But it doesn't mean that the main source only is mitochondria, because there are other kinds of organelles. But based on some experiments, we know some sources in plants more than animal cells. Okay, here are some equipments in our lab. On top in the corner, it is SLSPDs, which is actually some optical fibers and some probe. They're connected to some cryogenic stat. It's a fridge which cool down the sensor. The sensor is actually superconducting nanowire single photon detectors, or SNSPDs, and it is cooled to millikelvin, and it actually diminished the dark current significantly. So we have a high signal to noise ratio, and in the range of visible and also near infrared, it's a very strong detector. This is actually used also by other teams in Quantum Cloud Lab for quantum technologies. Here also, we are using that for photon detection. This is actually produced by JPL NASA for us. And we just received it just recently, and we still didn't start with the new device, but with the previous version, we have some results on brain tadpoles that it could record some signals. On the right side is just a simple demonstration that how these optical fibers can be located on the brain of a mouse, and then it is connected to detectors, which is located on photodetectors, and then it is connected to photon counters, and a time series data can be recorded. And on the bottom and the lower part is PMT, which is a famous one, which is the cheapest photodetectors, and it can work in room temperature, and even you can cool it down with some cooling systems to minus 30 degrees of centigrade, and it can give you real-time data at that time series. And the next one is an EMCCD camera, which is a high quantum efficiency. The quantum efficiency in the visible range peak is at 98%. And let me give you some examples, just some recent image we could get by this camera. Actually, this is the effect of anesthetics, is benzocaine 20%, and its effect on delay luminescence and UP in our lab. Why we are interested to apply anesthetics? Because there are some kinds of plants, they are sensitive to touch. For example, there are some plants that eat insects, or if you touch them, they move. or they are contracted. But when they just recently some papers are published, and they have shown that after the application of some anesthetic gas, it shows that their dynamic is deactivated. So when you touch it, they are deactive and they don't show no response. In this case, we're interested to see how this anesthetic gel or gas affects on the leaves, to help us to find of the bottom, the on-off bottom of the brain, for example, to understand the consciousness in the brain. So this is just a simple system in the leaves. When it deactivates some activities in the plants, why not to test it and find it out what is going on there. So maybe this kind of light emission can be some indicator and which help us to understand how it acts on some mechanisms. So we are interested to see what is its effect on delay luminescence first, and then what is its effect on UPE, and then try it with different techniques to find out the main mechanisms. Just with preliminary results, here we could see only the effect only on a small point of the leaf. We apply this anesthetic gel on the top row. If you look at that on the left side, this is after 10 minutes exposure, after 1 minute being in darkness. You see that that location of the that anesthetized doesn't emit light. But the rest, for after being 20 minutes in darkness, so the UP is started, and after 20 minutes exposure with EMCCD camera, we don't see that part that is stopped. So again, after 45 minutes exposure, after 20 minutes being in darkness, again, we couldn't see the footprints of this anesthetic effect. Again, after 70 minutes, we started again delay luminescence, but we could see the effect of this anesthetic gel. Also, we tried it, this back side and forward side of the front side of the leaf, that if maybe because of the refractive index of this anesthetic gel, it may be the effect, but we could see similar effects. The upper parts of the image are taken by EMCCD. Then we tried with ICCD, which is a kind of intensified CCD. EMCCD is electron multiplying CCD. So the mechanism of the detection is different. The quantum efficiency of EMCCD is around 95%, 98%, but the quantum efficiency of ICCD is around 30%, but it uses some kind of intensified mechanism that it can increase the signal to noise ratio. In the lower part, if you see, we applied this anesthetic gel on two parts, and after some time, after 5 minutes, after 35 minutes, after 65 minutes, and after 90 minutes, you can see that how these the area of application of this gel is expanded, is extended to a larger area after some time. So it means that it affected in some parts and it stopped this kind of emission. So it means that the mechanism that in delay luminescence, which is different with the mechanism of UPE, it shows that this anesthetic effect acted on the mechanism of maybe mainly on chlorophyll here. Also, then the left side is an image of two mice. One is dead, another one is alive. And this is for after 1 hour exposure. This is based on CCD camera, based on IVIS imaging system from our collaborators at Ottawa, but actually NRC. I mentioned here Ottawa, but actually they are from NRC, our colleague in this project on UPE. So you can see that when the mouse is alive, it emits light, but if it is dead, it doesn't emit light. And this is just the signal of life, this UPE. The right side image is my fingers. Some part of my fingers are the hydrogen peroxide are applied, and the rest nothing. You can see that after the application of hydrogen peroxide, the intensity of light is higher than the rest part. So this hydrogen peroxide can enhance the intensity of biophotons 1 or 2 orders of magnitude. This images taken by RMCCD after 30 minutes exposure in our lab. And also these are some other kinds of UP imaging in different countries. For example, in Japan, it can be used for diagnosis of cancer as a tumor in the body of a mouse. The intensity of that is higher, and also it can be used for metabolism variations in the body of human during sleep, and also adding some chemical agents to leave. On the bottom, you're seeing a face and hand. This is Roland van Wyk, the leader in UP research in Netherlands. And you see in the left side that this emission is taken by CCD camera, but with a spectral range in a limited range. And this is after a half hour exposure. And also some leaves at University of Arizona by CCD camera, Princeton camera. And on lower part, also from the human body and also from the plants and also human hands from Czech Republic, from University of Pulaski. Okay, this UP can have some applications. The main one is basic research, for example, quantum biology, and also can be some indicators to find out what is going on in a living system. And why we call it quantum biology? Because these are single photons, and a photon is a quantum concept. Also because we are using some quantum detectors, these for single photon detection. And in that case, we can take this topic into account as a quantum biology topic. And also it can be used some biomedical imaging tools used maybe to diagnose some diseases or some abnormalities in the living systems, and for diagnostic applications. And also maybe for photonic chip-based technologies for medical diagnostics or some, for example, BCI applications, it can be used. And also this kind of light can be used for food industry, for example, regarding vegetables. Some companies, they're using that. For example, in Germany, there's a company, they're using these for food industry and also agricultural applications. In Japan, there are a few companies, they're working in this context to find out the quality of and also the quality of food, and also the stage of the growth of the plants. And also it can be used for some kind of just artificial intelligence system design, and also real monitoring brain activity on Apple Watch. I will explain it in continuation. For example, one of the questions that can be answered in basic research is retinal discrete dark noise. It means that there are some photoreceptors, and there are photoreceptors in your eye and in the retina rather than cone cells. But sometimes in darkness, even they are located in darkness, they have some spontaneous firing. And the question is that why in an environment there is no any light, why they have firing? There are two kinds of noise in the eye. One is called continuous dark noise. The amplitude of that is 0.2 picoamperes. And we know that the source of this kind of noise is because of heat. But another kind of noise is called discrete dark noise. The amplitude of that is 1 picoamperes. And these peaks are exactly the same as the peaks that they are produced by receiving the single photons. And the question is that why in an environment there's no photon, there is this kind of firing? So in this paper in 2016, we explained that the rate of emission by UP and the rates of firing, they are almost in the same order. And maybe this UP is the main source because it's some endogenous light emission for cellular metabolism. And it can be a source and can explain it. There are also some classical explanation for this mechanism based on Boltzmann distribution in Arrhenius equation and also Hinshelwood distribution, but there are some discrepancies that it cannot be explained by these approaches. Okay, in our quantum cloud lab in University of Calgary, we are mainly interested in brain UP, and the question is that can we use this kind of UP as some, can we consider them as some signals, new kind of signals in the brain. This is still at the level of conjecture. We still don't know. They are byproducts, but they can play some informational role. This is the thing that we are interested to investigate in our lab to make sure. Professor Christoph Simon in our department several years ago, his group simulated that myelinated axons can act like optical fibers, and in different conditions after bending and even non-homogeneous cross-section, in all conditions they can transmit light with efficiency of over than 70%. This is still actually the level of conjecture, but it needs experimental confirmation. We are still working on that with different levels, but also there's a Chinese group leading by GIP Dai. They have performed some experiments, especially one published 2 months ago, that they experimentally have shown that there are some neural circuits in the brain that when you illuminate at some points, it seems there are different patterns of light propagation during brain slices. And they even could show that even single neurons can conduct light. And it seems that these neural axons can act like biological optical fibers. On the left side, here an image taken 2 decades ago by Japanese group for 6 hours. This is an in vivo imaging on mouse brain, and each image, there are 6 images. Each image is taken after 1 hour exposure. And they could show that the variations in the intensity have significant correlations with electrical activity of the brain, and there are significant correlations with EEG diagrams. And also some neurotransmitters applied to brain slices, and it could show that intensity of light increased again by performed these experiments by GIP Dai's group. So it may have kind of, if we consider it, even if it is byproduct, and even if it has no informational role, but it can be used for some functions, and it can have some futuristic applications. For example, one of the works we have done before last year is just a suggestion of new kind of technology for brain-computer interfaces. If you consider a photonic chip mounted on the interior surface of a skull, in the skull, it can make a sufficiently dark environment for this photonic chip, and it can detect these single photons in long term, even for several years as a long term, and it can be communicated to a smartphone or even a computer or even your smart wristwatch. And this kind of photonic chip can have different designs. It can be equipped with an interferometer for just in case that there's some synchronous activity of the brain during a cognitive task in the brain. So it may be because, you know, interference occurs between similar photons. And in that case, if there's some synchronous activity, it may create different patterns. And based on some machine learning tasks, it can be discriminated, and some features can be extracted as just something understandable by some algorithm on a smartphone, and it can maybe make a better spatial resolution, comparing with some of the approaches based on electrical activity of the brain. So there are 2 famous companies in this context. One is Neuralink by Elon Musk. They're trying to perform these experiments on the human brain. So Elon Musk is still following up the issue to get the permission to perform this experiment on humans, because he only performed these experiments on monkeys. But Synchron could perform on the chip-based technology on the human brain. So in this context, Synchron is one step behind, is forward comparing with Neuralink. So in this case, we think that photonic chip can be minimally invasive comparing with electrical chips, because these electrical chips, they're equipped with some electrodes that they should penetrate in the tissue of the brain. But for photonic chip, it doesn't need to even make injury on the meninge layers. it should be only mounted on the interior surface of the skull, and it can monitor. And also photonic chip can be faster, much faster, and also cheaper than electrical chips. So it can have some advantages, and we have discussed it in our paper in 2022. Also, some other applications it can have, for example, as a futuristic application, if we have a couple of photonic chips equipped, for example, with CMOS pixels and different locations, it can be connected to your smartphone or even your Apple wristwatch. And in that case, you can have some imaging after each 20 minutes or half hour. You can have an update of states, and it can give some information about the health state of your brain. And also regarding for diagnosis of some diseases, for example, like Alzheimer's, we could obtain some preliminary results that there's some significant correlations between Alzheimer's disease and reactive oxygen species, and also memory loss and the intensity of biophotons. In that case, maybe a photonic chip can help for diagnosis of Alzheimer's in advance after a long time recording of data on your small wristwatch. And another application, for example, can be some sensors on your skin, because there's some research on the health state of the hands. If you look at it on top, there's some image of hand. The first one on the left side is the image of hand under room light. The second one is normal UV emission. The third one is after the application of zeaxanthin, so the intensity is increased. The next one is hydrogen peroxide, and the last one is after the application of phantom. And in different papers, the people have shown that there are some correlations between the health state and also the intensity of emission from dorsal and palmar sides of the hand. In this case, maybe it is possible to use a wearable sensor equipped with photonic chips to diagnose this kind of photons as some application. Okay, now I'm in the last slide. Actually, we intend to hold a quantum biology conference in next year in Calgary. It has a beautiful nature, and perhaps it will be held in Banff or maybe in city of Calgary. So stay tuned regarding the announcement, and hopefully we will see you here and have fruitful discussions together. Thank you so much.
Thank you very much, Vahid. It's very cool to have you here now. It's a pity that we won't be able to see you in person next week. And this field of biophotons is really, really cool. So we are running slightly earlier. Does anyone have questions to Vahid? And actually, Vahid, can you see the question and answer? There were some question and answers in the Q&A box.
Oh, okay. Let me check the chat box.
Not the chat box, but the Q&A box.
Q&A box. What is Q&A box?
I
don't see it. Please.
Are you co-host? Yes, you're host. So there is. Let me read that to you. So there's one question. I think it's talking about delayed fluorescence, right? I guess I don't understand what's the significance of that delay. I understand exposure duration for collecting more photon counts, but what's the significance to start the recording? I'm not sure if that makes sense, and I think it has to do with delayed fluorescence.
I can actually,
yes, go for it. Go for it.
Yeah.
Yes.
So there was delay of 5 minutes or 27 minutes, and after that you started recording. So delayed fluorescence probably, as you had mentioned, might be the reason why you start recording 4 UPE later. So if that's the reason, the time equal to 0 is when you stop the all photon stimulus. Is it what it is? I mean, what sort of darkness are we assuming here?
Actually,
which frequencies have been cut off?
Actually, I'm not sure to understand your question, but if you are talking about prompt fluorescence, not delay luminescence, the prompt fluorescence, we could record it because it needs careful detection, because it happens on the first nanoseconds, because it takes for us, we have a big tent, a black tent, is actually our dark room, is a big size, and our EMCCD camera is located there, and the sample is under this EMCCD camera. First, we illuminate it with some flashlight for a few seconds, and then we put it under this EMCCD camera, and then we go out of the tent, and then we close it, and in the dark room, so it's the total darkness, and then we start recording, maybe after 1 minute being in darkness. So what we record here is only delay luminescence, and the mechanism is different. But actually, we were not interested in delay luminescence because I think this mechanism is known fact, and there are many papers published regarding delay luminescence and the mechanism, what is going on. This is photosynthetic based, and the main source maybe because of photosystem 2 and something regarding happening in chloroplasts. But here, we are mainly interested in UPE. But after the application of anesthetic gel, we could see that its effects can be seen in delay luminescence, not in UPE. So then we were interested to investigate delay luminescence to see why it is deactivated in delay luminescence, but the effect cannot be seen in UPE. And why? Because the mechanisms of the sources of these emissions are different. That's the reason.
Great. I mean, just to follow up on that one again, when at time equal to 0, you said you flash the thing at something, and then when the light is gone, that's time equal to 0. And but the flashlight has some spectrum, whatever that spectrum was that was illuminated with.
This is white light. This is white light. This is a simple one. And also we have some recordings of different, because we have different kinds of laser sources with different wavelengths. So I didn't talk about the responses, but the responses are quite different. And also, so it seems that delay luminescence is wavelength dependent of the illumination source. So in that case, yeah, that's a good question. And it seems that also the intensity and also the duration of this emission will be different. And it depends on the wavelength illuminated. So we have TISAF laser. You can change the wavelength of that for each 10 nanometers variations. You can see also the variations on delay luminescence. We still didn't publish our results because we're still working on that to make sure about the details. Then maybe another meeting.
Changing your laser by a couple of nanometers gives you different physiological responses?
We just tried with 50 nanometers difference, and we could get different responses, and
that's
we could
amazing.
record it. Yeah, but with 10 nanometers, we still didn't try, just because we were only curious to see if the effect can be different. And in that case,
I mean, 50 nanometers, what's the spread in energy there? Because I would say that 50 nanometers
for example,
temperature is I mean, it should.
No, it's a laser. It's laser wavelength. As because of, for example, TISAF laser, you can.
I know, and you're that I get it. And you're saying that by moving this laser by 50 nanometers, I don't know how much that is in.
No, changing the wavelength, the wavelength of the laser. So first we tried 600, and then 650, then 700, then 750, then 800. Yeah.
50 nanometers. How much in milli-electron volts that is? I'm thinking because.
Yeah, like in the visible range.
Thermal energies is like 25 milli-electron volts. So all I'm saying is that I just want to make a comparison, because at room temperature, 50 nanometers might be okay. 50 nanometers might be 50 nanometers. You can change color. Okay, fair. Okay, got it. Okay.
Yep.
Cool.
So no question anymore.
So, what do you think is next? I mean, so I have a question that is like a little bit of speculation, but Vahid, we've talked about that before, right? all those phenomena that you're seeing are related to photoreceptors that exist in cells that are not usually exposed to light. It's not like skin cells, it's not like photoreceptors in the eye. There are photoreceptors in the cells, like inside.
Photoreactive proteins. Maybe it's better to say photoreactive proteins, not mainly photoreceptors. Yeah.
Well, it's proteins that can absorb photons.
Yeah, or just show some reaction to photons. Yeah.
What's the difference?
Yeah, maybe this, absorption, and also, yeah, maybe it can be seen, it can be said that, but maybe the experts in this context are careful to use this kind of terminology. That's why we first use photoreceptor, but people say that, okay, photoreception, maybe the mechanism is something more fundamental. So you should say photoreactive proteins. And in that case, maybe it can be use some biological antennas. Yeah.
Okay. So you think that those things are there with a function, that those photoreactive proteins, it's not that they didn't fall out during evolution, that they remain there for a reason.
Yeah, this is a really good question. For example, there are different kinds of photoreactive proteins in the brain, because we only know the photoreceptors in the eye, which responds to external photons, and we perceive it and we see the environment around us. But there are also some photoreactive proteins also in brain, and not only in the brain, in most of the biological systems, they exist. And the question is that why they exist after the evolution. So there should be something inside living cells that they could respond it and they could survive. And that's why that maybe we did this conjecture that this endogenous light emission inside cells and is UP may be the main source. For example, based on the discussions with Alina, if you remember, for example, just a few days ago, one paper published in Nature by Alina and her supervisors group, again, finding a new kinds of photoreactive proteins and bacteria. They are hunting new bacteria in deep oceans. And the sunshine can reach only to the depth of 100 meters, but after that, so it cannot penetrate more. So in deep oceans, it's a dark environment, and there are different kinds of bacteria that are equipped with photoreactive proteins, and also they are equipped with rhodopsins, this photoreceptor. And the question is that why they exist and how they can use it and to absorb light and make food to survive. And also when we discussed it regarding the existence of this kind of endogenous emission, this spontaneous emission, so they were very interested to know more about it. And that's why we are working on that review paper on dark activation of reactive, molecular reactive proteins. Yeah.
Exciting. Final questions to Vahid.
Wow, this is fascinating. It's the first time I've heard this, but I'm excited because what if there are other spectra, parts of the spectral electromagnetic spectrum that we have receptors for inside the body, which we haven't probed. So I wonder if we can develop sensors for that, and like your techniques will be translated to those spectra.
Yeah, maybe that's a good question, but we still didn't investigate that. But still, also we are working on this photoreception mechanism and this photoreactivity in biological systems. So we have hired a person who is an expert in this context. Then let's see what we can find out. And yeah, that would be a nice research field to explore what is going on with photoreceptor and also photoreactive proteins in living systems. Yep.
Exciting. Thank you, Vahid, so much. Maybe we take the next 2 minutes to say again that Susanna prepared a very nice feedback form for us. Please go there, spend 2 minutes. We're taking any feedback, constructive feedback that you want of like how we can do this better the next time, and what you enjoyed, what you didn't particularly enjoy. Please go to that Google Forms and leave your feedback to us. Thank you so much. So I think we could move on to our next speaker, Jonjo, who's already there. If you do not know, Jonjo is one of the authors of this famous book that we keep making a reference to called Life on the Edge. Go there and buy the book. Let me Nathan, can you find the link to the Amazon site of the book? It's
Sure.
Probably the best layman's book on quantum biology. Jonjo and Jim, they describe several flavors of where quantum mechanics might be at play in nature, and it's an excellent introductory book. So Jonjo, it's super pleasure to have you here with us, and also because you are the director of of the first quantum biology doctoral training center in the world. So we would like to hear a little bit about what has happened since the first ever quantum biology doctoral training center was created at the University of Surrey, how you're taking this forward, what are some challenges, what are some delightful surprises that you've had along the way, and anything else you would like to talk about. And I think that after about 30 minutes, we plan to open up for discussion so that we can talk about education in the context of quantum biology.
Yeah, thanks, Clarice, and thank you for a very warm introduction. And yeah, so quantum biology and the teaching of and how we got there. So now, let me see. The first thing I wanted to mention was how one of the ways in which I came into thinking about the difficulty of quantum biology was actually writing the book, Life on the Edge. And when Jim and I got together to write this, we both had our expertise, obviously mine in biology and Jim's in physics, and we wrote our bits. And I couldn't really understand what Jim was writing, mostly, or a lot of it, and he simply couldn't understand what I was saying often. But we'll both take the approach that, well, Jim's a very clever physicist, so he knows what he's talking about, and I'll leave it to him. I don't know any physics. And Jim took the same approach in the reverse direction. John knows his biology, must be right. And only really when it came to our agent, who read them both and said, I don't understand either of these, did we really realize that there was a problem. So what we then went on to do was every part of the text that Jim had written, I would rewrite to make sure that I understood it in a way that made sense to me. And then Jim would correct the things which I had got wrong. And similarly for the biology, he'd rewrite my biology to make sense to him and get rid of words that he didn't understand, etc. And eventually banging that back and forward between us, we got a book that hopefully is comprehensible to both sides of the equation. But it did teach me how difficult it is to communicate between the disciplines. And let's see if I can share my screen. So I've prepared some slides. We had some confusion at the time, so I was leaving another hour to prepare some slides later today. But let's see if I can. Okay.
And the time issue is totally my fault because the US changed times this Sunday and I hadn't realized that this would impact the international speakers. Sorry for that.
That's quite all right. Right, so here we go. Hopefully you're all able to see the first slide. Yeah?
Yes,
Teaching
but not
quantum biology.
Yes.
Good. Okay, so that's what we're going to be looking at. And actually another experience prompted my thinking about this problem when we were putting together. So anyway, as Clarissa said, we founded the Quantum Biology Doctoral Training Center at the University of Surrey back in 2018-19. And and it was funding from the Leverhulme Trust. It's one of the problems in quantum biology, finding the right funding agency. And this is really more to do with research, but it is worth thinking about for a moment because it is difficult. The Leverhulme Trust has the rather peculiar criterion for its funding that they won't fund anything that could be funded by the major governmental and big charity in the UK. So that's the Biology and Biotechnology Council, the Engineering and Physics Council, or big charities like the Wellcome Trust. They won't fund any of that. So that immediately makes them a little bit out on the edge, and they will fund more interesting stuff. But it is a problem getting funding in quantum biology because you have to convince the biologists, and they're skeptical of quantum biology, of any quantum mechanics involved in their system, and you have to convince the physicists and engineers, and they're skeptical about the biology. So it was a challenge as well. But anyway, these are the different kind of components that we said we were going to pull together in our quantum biology doctoral training center. And unfortunately, the Leverhulme gave us the money, and we started recruiting in 2018. And you can see on the top left, the students, number of students that were recruited in each year. And the picture on the bottom left shows a year ago on the Surrey Hills, the cohort we had at that time. Here, the top right picture shows some of our students. You're going to be meeting some of our students in the meeting. They're not attending today. Most of them aren't attending because they're actually heading over to the US as we speak. But you'll be meeting quite a few of them at the meeting. And here's an example of quantum biology, the picture on the top right, which doesn't make any sense at all, because actually the three students that you see, Sapphire, Louis, and Les, they're all theoreticians. So them in a lab coat is totally inappropriate. And it looks like a microwave there, and that's probably the closest they get to using an instrument in the lab would be a microwave oven. But that is the issue with quantum biology, that we have such a large range of expertises that go into the science. Now, let's see. As I said, I haven't prepared all of these slides, so I can't remember. Okay, this is a joke. So anyway, I'll give you a moment to read the joke, and then okay, so it's kind of interesting. It's a kind of men are from Mars, women are from Venus kind of a joke, which is saying that men and women are very different. Whether that's true or not, physicists and biologists are pretty darn different. I thought this was kind of good because women are weird because they can both be happy and unhappy at the same time. That's kind of quantum mechanical, and then men can too. So yeah, we can. So I'm taking this joke as kind of between a biologist and a physicist, and that a physicist is saying, well, men and biologists can, or particles can be in two places at once, and biologists saying, not in my experiments, they can't. But really, we're trying to convince them in quantum biology that yes, they can. Particles can have these peculiar features. Now, I won't. There's another punchline graphic on the next which I won't show the caption for because it's a little rude. But you can guess how you can have a guess at how you can make somebody happy and unhappy at the same time. How his wife, presumably husband, wife sitting here, how they can be, how the husband can be made happy and unhappy simultaneously. And you can think of what the caption might be in that last slide, but I won't put it up at the moment. So, and this is one aspect of the culture. This, everyone knows this joke about the spherical cow. That in biology we deal with lumpy cows, wet, lumpy, and living cows. And when it comes to a physicist looking at the problem, well, let's imagine a spherical cow. And that kind of culture clash is one of the things that I want to explore for a moment. There we are, very different in the way that biology is taught, the way that biologists come into biology, and the way we think. This isn't always true, and it isn't. Maybe it's something like 60 or 70% true that biologists tend to think in pictures and in images, and physicists tend more so to think in equations and mathematics. Not all. I remember reading, if anyone's I read the biography of the Nobel Prize winner, whose name will come to me in a moment, but he used to imagine equations as real physical things, real physical objects, and then he could encounter problems with the equations. Who am I thinking of? The guy who came up with electrodynamics. Someone help me here. Nobel Prize winner.
James Clerk Maxwell.
Sorry, no, sorry, quantum electrodynamics.
Feynman, Schwinger, and
Feynman's biography. If anyone wants a pretty good read in popular science, biography is Feynman's biography, which he tells this story of how he thinks. Nevertheless, he was a very good mathematician, but he thought in pictures, and when he saw an equation, he would think it out and say, okay, it's going to twist here, turn here, no, it's going to come back, no, that can't be right. So it's not entirely true that all biologists only think in terms of images, and physicists only think in terms of equations, but there is that kind of tendency. Now, okay, so, and theory, the role of theory is very different. So biologists, actually, go back to saying a little bit more about this. So we think, as biologists, we tend to think in pictures, and physicists in theory. And that's partly because a lot of biologists, at least in the UK, maybe the same is true in the US, I don't know, that they kind of flee to biology because they can't do the maths. And that's partly a consequence of often very abysmal mathematics teaching in schools. It's in the UK, it used to be pretty darn awful. I think it's improved in recent years, but it means that a lot of students get scared of mathematics, and then they're still interested in science. So what do? They do biology, which means that they're often ill-equipped to then take on quantum biology, which has mathematical aspects. So that's another aspect that we have to, a potential problem we have to deal with. And then the role of theory is very different, I think, in physics and biology. There isn't much theory in the sense of a law, say, in biology. The closest I think we get to it is probably natural selection, which Charles Darwin and Russell, Wallace came up with in the 19th century. That's as close as we get to something like Schrodinger's equation in biology. It's often said that Dobzhansky, the Hungarian-American biologist, said nothing in biology makes sense except in the light of natural selection, in the light of evolution, and that is still kind of fundamental to biologists, that if it doesn't make sense in the light of evolution, there's something wrong. And that's as close as we have to a fundamental theory of biology. There's no kind of effort to make a unified theory of biology, as there is a unified theory of physics. So there is that kind of cultural difference as well. There's also a difference in language. And this came to me when I was having a meeting, putting things together for the Quantum Biology Doctoral Training Center. And we started, we were about 12 or so of us were in a room and trying to pull together ideas for what would go into the proposal. And a biologist was talking about a particular phenomenon. It doesn't really matter what it was, but the physicists then ask, well, how fast does this happen? How fast is this phenomenon that you're looking at and you think might have quantum mechanical aspects? How fast does it happen? And the biologist said, well, it's very fast. The physicist says, what do you mean very fast? What's very fast? Well, it's very, very fast. And then eventually they pinned it down to units, and he said, well, seconds. And the physicists all fell around laughing, because seconds, fast, even terms like fast mean very different things to the different disciplines. So that's also a challenge. And what I wanted to do, Clarice, you might be able to answer this. How many of our audience today are students?
I would say about 80%.
80%. Okay. Now I'm gonna ask for some audience participation here. What I'm going to do is I'm going to ask one of our. I'm assuming that the audience includes both biologists and physicists. Is that a reasonable assumption as well, Clarice? Do you think?
Yes. Yes.
Okay. Now I'm going to ask some volunteers from the audience to explain ideas from their discipline to the other discipline. Okay. So we have on here, of course, Schrödinger's equation, and I'd like a volunteer from a physicist, one of our physicist students, to explain what this equation means to a biologist. Would anyone like to take? You could put your hand up in amongst the participants.
And Jonjo, sorry, but I will volunteer people if they don't willingly help here.
Okay,
I am familiar with them. Yes, no, go for them. Who wants to do that? There's plenty of physicists in the audience.
Who can explain Schrödinger's equation to the biologists in the audience? Something in the chat line here. Is that anyone? No, Feynman lectures. Okay. Do we have a volunteer, or do you have to bully someone into the getting in the hot seat, Clarice?
Oh, I will. Leo, you knew it was coming for you. Leo, can you help us here? Leo from Brazil.
No, for sure. Sorry, I have no clue about it. No.
Okay, there must be a physicist student who's brave enough to tell us, biologists like me,
oh,
what this equation
Irina.
means.
Irina is there. Internet's too bad. Okay, we're running out of physicists.
Okay, well, while the
Ebba's out.
physicists...
No, one last try. Sure. Ebba's out. Come here. Okay.
Okay, I can try.
Okay.
Well,
and now, before you start, sorry, what's your name?
Abasalt.
Abasalt. Okay. Before you start, Kuri, what I'm going to ask is for those biologists who don't know the answer to this, I think this would be every biologist. What I want you to do at the end of this is to put your hand up whether you understand the explanation or not. Okay? So be prepared to do that at least. Put your hand up for all the biologists in the audience, whether they, and that can include the non-students as well, if you wish to. How well your knowledge of the Schrödinger's equation has been improved by what you're about to hear. So off you go.
Sure, I will try. I will do my best. When we go to quantum mechanics, like we deal with really smaller size particles like atoms and so on. Here, okay, the mechanic, how we can describe their evolution, their motion and everything is different than classical mechanics. So we cannot just simply use Newton's law here. So then we use quantum mechanics, and quantum mechanics are most reliable parameter to define things and to study these particles is wave function. And what does this Schrödinger equation mean? Is actually, it is relating the evolution of this wave function of like any system, can be atom or electron and anything, it is relating it to the energy of the system. So basically, it says that how this wave function changes in time in response to the energy of the system. So on this equation that we are seeing now on the screen, so the left side, it is actually the time derivation of the wave function, psi is the wave function. This h bar is just a constant, which describes this, the whole thing is quantum. So on the left side, you have this time evolution, and on the right side, this h means in physics, we call Hamiltonian. But when it acts on a wave function, at the end, it gives the energy of the system. So this function, this equation very beautifully, like if you imagine, like you want to understand how the system evolves in time with response to like its energy. So it is relating the energy and the time evolution of one system.
Okay, is that the end?
Yes, I guess.
Okay. Okay, now if we look at participants, I'm just asking those participants who can, I'm not sure if I can see all the hands going up, but those participants who are biologists, tell us if you now understand Schrödinger's equation. And we're going to have 2 votes. Yes, you do, or now you don't. So first of all, who understands from the explanation we've just had Schrödinger's equation? Okay.
There's a thumbs up. There's a thumbs up. Are we
One
going
thumbs
to...
up. Okay.
2 thumbs up.
2 thumbs up. Okay, good. And who doesn't still from, so I'm asking, you've got to vote one way or the other. Who still doesn't understand Schrödinger's equation? So the people who had their thumbs up, take them down. And now we'll wait for the other biologists to vote one way or another. So put your thumbs up if you still don't understand Schrödinger's equation. Okay, let's see, 1, 2, 2. Okay, any more? So it's 2 votes for, 2 votes against, I think. Is that how
and then
you
there's
see it?
there's 2 comments from the chat, one from myself, the other one from Nathan, saying that we as physicists we have never understood it.
Well, I suppose there is that, you know, Bohr, I think, famously said that anyone who says he or she understands quantum mechanics doesn't understand quantum mechanics. Yeah, I think it's still a challenge to get your head around it for most biologists, more so than it would be. At least you would understand how to use it, Schrödinger's equation, whereas most biologists wouldn't have a clue. But let's see if we can get biologists. Now, we've got on the other side, the origin of the species. That, I think, is too familiar, evolution, everyone knows that. But would a biologist, one of our biologists in the audience, be able to explain the difference between transcription and translation to a physicist? Does anyone want to volunteer to do that? Put your thumbs up if you'd like to volunteer.
I can go, yeah, if no one wants.
Okay.
Yeah, transcription is basically produce mRNA, and translation translates this code from mRNA, from nucleotides to a protein, basically, just codifying information.
Okay. Now that's a nice simple explanation. How many of the physicists in the audience understood that? Who didn't? If you knew about it beforehand, please don't vote. But if you didn't know what it was about, so Susanna, I can see now understands difference between transcription and translation. Okay, anyone else from the physicist side? Do you understand? Okay, how many still don't understand the difference between transcription and translation? Certainly one. Clarice has voted no. And Sandro, co-host. I mean, I guess some of it is just that we use terms. I mean, I've forgotten who did give us the explanation. But terms like mRNA, what's that to a physicist? Most physicists don't have a clue. Very familiar terms. So we tend to use very familiar terms, and that can be part of the problem. But we also have this huge body of knowledge that we've absorbed over the years, both in our education, but also in our interests, that we absorb and take on board. And we can talk we tend to associate them, at least academically, when we're talking science, with people of the same discipline who speak the same language. And it's tricky then to communicate across those borders. So what we try to do then in the, let's see what, my back, I'll stop sharing. Yeah, I think no, I do need to, okay, my slides are no longer moving forward, so I'm going to stop sharing and then do it again. That usually cures the problem. Okay. Okay, so you can now see the slide again, hopefully.
Not in presenting mode, but yes.
Okay, there we are, presenting mode. Okay. So yeah, so those are the problems, I think, and we've been trying very hard to deal with that. And we did think, okay, well, we recruited from both sides of the divide. We tried to get a fairly even number of biologists and physical sciences, so engineers, mathematicians, physicists. And in the first year, as you saw in the earlier slide, we had eight students. So we thought, okay, well, how are we going to get them to work in the quantum biology field and to understand each other and communicate with each other? And first of all, we thought, okay, well, I could teach the physicists some biology, and Jim could teach the biologists some physics. But then we had a better idea, better from the perspective of Jim and myself, because we didn't then have to do much, is we got them to teach each other. So, for example, this is Andrea Rocco, who's one of our quantum physicists. He was in charge of say, putting together the quantum biology program. And what we had then was, for example, the physics students were teaching the biology students, and these were on the physics side, and these were on the biology side. And from the difficulty, as I mentioned, with teaching quantum physics to many biologists, is that their maths isn't that great, and that includes me. You know, basic calculus I can get my head around, but more advanced calculus is not something I can do or even understand fairly easily. So we had to we had our physicists teaching basic calculus to begin with, and differentials, integrals, simple ODEs. And then biologists teaching stuff like chemical reactions, rate equations, steady state stability, gene regulation. So we had modules on each kind of topic, and then another module later in the course. This was all taking place during the first year of their arrival into the PhD program. Remembering the PhD program in the UK is only 3 years, so things are a bit tight. And then back to physics with Newton's equations, energy, momentum, Hamiltonian's and Langevins equations of motions, and then introduction to quantum mechanics, where we looked at the double slit experiment, the mathematics of quantum mechanics. So, for example, complex numbers, yes, as a biologist, you kind of know that they're there and have an idea of what they are, but they don't really play much of a role in biology, so we don't really know them very well. State spaces and stuff like this, we're not familiar with. And then Schrödinger's famous equation, of course. So get to grips with that, and then hopefully solving Schrödinger's equation for a free particle and the particle in the square well. So getting the students to teach each other was, I think, very crucial because it forced them to communicate. They were doing the kind of things which I've just asked you guys to do, which is, a physicist to explain to a biologist what the Schrödinger equation is. And what you quickly find is the terms that they were using don't make any sense to the biologists. And similarly, in the other direction, when the biologists were saying, discussing transcription and translation, they say, well, what's this mRNA business? And then you have to go back and explain what messenger RNA is, which what it stands for is, and it really forced them to go back to the fundamentals and then build up from those. It's a very different experience to teaching from a base upwards. You have to start and then go to the base and then back up again. So that was a good experience for them, but it did force them to learn how to communicate, and it's back to that femtosecond example I gave, to know what fast means in a physical sciences sense compared to a biological sciences sense. And there's still part of the problem still remains. There's still a gulf between the biologists and the physicists, but it is lessened, and they can both, I think, and you'll meet several of our students when they turn up pretty soon at the meeting in Galveston. They at least can talk to each other. Now, I'm not sure. I think that might be my last slide, although I've got a feeling it isn't. Oh, yes, here we go. Oh, no, it isn't. That's right. Okay. And this just tells the diary, but you don't really need to see that. Okay, so that's the basic thing that I came to talk about, the challenge of quantum biology. And it's an important challenge because I'm convinced that whatever happens in quantum biology, there will be a fusion between physics and biology, and we're going to have people working. A big part of 21st century science will be people working at that interface between quantum mechanics and biology, just as biochemistry was essentially a fusion of 20th century biology with 20th century physics, well, mostly with 19th century physics, actually, classical mechanics. 21st century biology will have to fuse with quantum physics, and I think whatever our understanding of quantum biology is going to be, that fusion will happen. So I think that's the only thing I was going to say, except to say that there's going to be the Quantum Effects in Biological Systems meeting at the University of Surrey this June, at the end of June. I hope to see many of you at that meeting. There's the website for it. You can register for the meeting right now, and I hope to see you soon. I think that's all I was going to say, but I'm happy to hang around a little for discussion if would like to. Stop sharing now. Okay.
Oh, thank you, Jonjo. So what are your questions to Jonjo, specifically about the training of future quantum biologists? I have plenty of questions and a proposed question for discussion.
I have a question.
Yeah, sure.
So what kind of areas of the workforce, other than further research in quantum biology, which of course I've been able to continue in, but if someone is doing quantum biology as a PhD, what other options might they consider other than ongoing work in quantum biology research?
Pharmacokinetics will be the first obvious one. I mean, the interaction between a drug and its receptor is an interaction between one molecule and another molecule. There's no way that doesn't involve physics. So, and the physics it's going to involve has got to be quantum physics. A biology is hitting quantum mechanics, whether it likes it or not, when it's recognized. One of the features of biology that we are discovering is that single molecules or small number of molecules make a difference in biology. And that's what makes biology different from, say, chemistry. Small molecules, small numbers of molecules that really make a difference in most chemistry. But because of the amplification of effects in biology, they can make a big difference in biology. So a lot of biology is going to the level of examining single molecules and the motion of single molecules, dynamics of single molecules, and areas like pharmacokinetics. It's one molecule versus another molecule, and that's certainly an area that is going to involve it. But things like vision as well. We know that vision is got to be quantum mechanical at some level. You have photons hitting a receptor and causing a dynamic change. There's no way that a physicist would say, no, that's not quantum physics. It's involved photosynthesis, of course. So lots of areas, I think. But I would say the problems, for example, in protein folding is fundamental. It's where proteins are a string, and then they fold into a particular configuration. And that particular configuration is very particular because it generates all of the properties of the protein. And most proteins are enzymes, and they perform this fantastic feat of accelerating enzyme chemical reactions by many million-fold. And they do it because of the precise location of all of their amino acids. The proteins are made of amino acids, and inside the enzyme, they are precisely orientated with respect to each other. And that is certainly, but now there is a computer, of course, now, I think, what's it called? Anyway, there's a supercomputer which can with very, very good efficiency predict an amino acid, a protein structure from the amino acid sequence, but only by kind of taking all of the structures that are there and doing a kind of, okay, it looks a bit like that one and a bit like that one, so we'll take something in between. But there's no way of going from the fundamental string of amino acids that make proteins to doing it in a fundamental way. But that process of folding a protein is also fundamentally quantum mechanical. A single molecule interacts with itself and forms a particular structure. So understanding protein folding is going to be a quantum mechanical challenge, whether its counter is quantum biology or not. So, and that goes fundamentally to structural biology, which again goes into pharmacokinetics and pharmacology. So I think there's a lot of potential areas where quantum biology can play a role.
So you're saying, and I think I've heard this before, that the kind of picture we have, this toy model of the protein folding down this thermodynamic landscape in a semi-classical picture, it's really not adequate to resolve Levinthal's paradox.
I can't, I mean, no one's really, because they're so complex, it is the spherical car problem, so complex, no one's ever attempted to try to take 100 amino acids and find, okay, what is the most stable structure? Because you assume that the molecule goes into some kind of stable structure, probably not the most stable, but the first one in some kind of landscape of possible structures that it falls into. But it does this with extraordinarily efficiency, very quickly, and the best computers that we have can't predict that structure except just by doing it from a cut and paste kind of approach. And yet a protein manages to do it in millisecond times. So how does it calculate all of those energies of possible structures that it could go into and find the right one? To follow how it does it must surely take equations of quantum mechanics. It's because it is, again, single particles interacting with each other. That's what's delivering that final structure. So yeah, I would say it's got to be something that classical approaches are going to fail, and they do so far.
Doug.
Okay. Hi. So I have two questions. One's a specific one, one's a general one. So, so John Joe, are any of the course materials that you guys developed available for other people? I found particularly attractive that you treated the the Hamiltonians and Lagrangians in the classical sense, because that's a real barrier. And part of the reason it's a barrier to the biologist and probably even the physicist is that even in physics, it's taught as a calculational trick, and it rather than basic understanding. And it turns out that even in 1848, Jacobi, as in Jacobian matrices, was writing a textbook, and he was complaining about how Lagrangians were treated as a mathematical trick rather than any understanding. So if you guys have some insight on that, that alone would be great, but it would be nice to see the whole program.
I will get in touch with my colleague Andrea Rocco and see if he's happy to share the stuff that we haven't pulled it together into it was done more or less on the fly, but I'll see what we've got.
Okay, and then the general question is, did this approach seem to solve things? You know, the biology teaching the physicists and vice versa. And the reason I asked that is I've long wondered whether this is a left brain, right brain thing, and that there's a deep problem with the barrier.
Yeah, it certainly reduced the barrier, I think. It's not quite quantum tunneled through the barrier, but it's reduced the height of the barrier, I would say. And we're certainly able to communicate between our physicist students and our physicist students better now in their third year than when they arrived in their first year. And now can understand, you know, biologists can sit there and listen to something about Schrodinger's equation and get a reasonable idea of what's going on. And to show how useless I am at physics, I'm going to have to rush out and get a power cable because I've just seen that I'm nearly out of power. I'll only be a second. I have some more.
So Clarice, do you find that your students can talk to each other after a while? The physicists and the biologists?
We don't have biologists yet. We have a physical chemist. So we're starting, I mean, we're a bunch of quantum experimentalists, and we are learning from the from the physical chemist, and the physical chemist is teaching us about proteins and things. We haven't had a pure biologist yet, only some undergrads.
So
yeah, can I ask, for example, Alessandro, that's there. Alessandro, how's the communication been going in the lab?
No, pretty good. As you said, we don't have a real biologist, and we're still thinking mostly about the quantum stuff. So we didn't go through that hardship yet, but yeah, I feel we're trying to learn a lot of the bio dictionary.
So
to try to understand. Yeah.
When I switched from physics to biology between undergrad and grad, over the summer, the way I taught myself biology was with Isaac Asimov's Intelligent Man's Guide to the Sciences. Sorry, it was Intelligent Man's, but it was actually quite good. And it was amazing to me to find out that all of life is made up of basically 4 kinds of molecules, you know, proteins, fats, sugars, and something else, and other stuff like that. And I'm sure nothing in it is incorrect now, it's just that we know more.
Cool. Can you post maybe a link in the chat or something?
Oh,
like
okay. Yeah.
I think, yeah, and like, I think maybe it would be cool if on the biology side, if you guys have like resources for beginners, maybe to post it in the chat, and maybe the physicists can do the same for the quantum stuff. That would be cool. I started doing this course on biology, molecular biology. I can find it and post it in the chat. Maybe.
So I was just from my own experience with interacting with biologists, I'm a physicist, and we discussed a lot of crazy ideas with my colleagues who are biologists. And I taught in a classroom which had undergraduates from physics, chemistry, biology, mathematics, and we had to teach them nanotechnology or something like that. And the problem that we all saw, that was the language and the questions when they ask, what is the level of satisfaction of the answers when the answers are given? So if somebody says, as you have clearly pointed out, what is the time scale? What's fast is fast. The perception is different. Similarly, languages like evolution, time evolution versus evolution of species, that word kind of gets misrepresented. And so just being careful about the jargon in the classroom, or hyper aware of, this is a jargon itself, has helped my students to sort of recognize and when to help each other. We break out into small groups, as you have clearly said that, peer teaching helps. And I think this is also true with peer learning for anybody who's at any level. It just not have to be students, but us, as researchers also, we need to just sit down and teach each other fields. And I think that's the way to go forward.
One of the most important things, I think, is for people to, for students and everyone, I think, to recognize and admit to not understanding stuff. And that's, I think, often the greatest barrier, that people are sitting in a lecture and it's just going straight over their head what's going on. And really the lecturer should be told at that point, look, you're not making any sense anymore to most of the class. And I think students have to, in this difficult area of quantum biology, we have to be more assertive in both directions. Say, no, stop, tell me what an mRNA molecule is, or tell me what you mean by superposition or whatever else. And I think we do have a particular difficulty, I think, in going forward in the quantum biology field, in that there hasn't been, I think, a clash of disciplines, or a coming together of disciplines so diverse at least in my lifetime, where a productive research field requires people who are so different in their approach to science. And I'd be inclined, at least or even in research meetings, in quantum biology, of course, everyone has to go to a certain extent in their own discipline, and not everyone will be able to follow. But we should have, at the end of a talk, or at key parts of a talk, that you have to explain it to the other side. So if you're a physicist, you've got to say, okay, what this means, okay, what this equation means is this, and explain in words that a biologist can understand. And similarly in the other direction, a biologist has to say, okay, this is important because this and this and this. And I think if we had that attitude, that it would slow things down, because I know lots of talks that I felt, okay, you've lost half the audience here now. And it's kind of would be tough to say, look, stop and rewind. And it would make, but I think it would make people really kind of unpack their own ideas a little bit clearer, in a little bit clearer fashion that would help everyone. But yeah, I think we have a particular challenge in quantum biology because of the clash of very different disciplines.
I have also another question, which I think, Susanna, you actually raised your hand. Go for it.
That's all right. Do yours first, Clarice.
Thank you. So the field of quantum biology has a lot of baggage, right? Historical baggage associated with it. And a lot of people learn about quantum biology, that quantum biology even exists, because of weird new agey claims. I'm sure that you've heard people thinking about, oh, quantum is how we use telepathy, or things about like quantum consciousness. So I wonder if what we can do to improve this bad rep, and to say that there are people who are seriously thinking about quantum effects in biology, and what we can do as a community in order to dispel these nefarious baggage that some people associate with quantum bio. For example, Jojo, I don't know if you have, so during the pandemic, our quantum biology center here at UCLA, we organized something, an interdisciplinary workshop that was dubbed a square table, that brought people from biology, physics, chemistry together to discuss. And the people who participated in this seminar were friendly people, right? People who took their time to discuss about quantum biology. I had one of my students told by someone at that document, at that workshop, that I was irresponsible for having them work on a quantum biology project for their PhD, in the sense that someone who actually donated their time to be there to engage said that it was irresponsible for them to be in a quantum biology PhD program because this wasn't a thing and this could harm their career prospects. I wonder, Jojo, if you, and everyone else here, if you have, is it just a matter of rebranding in the same way that cold fusion is now called low energy nuclear reaction? So is it just a matter of rebranding? Should there be more PR to say that, well, there's serious people who do not care about consciousness, who do not care about telepathy. Actually, we're doing good science. Yeah, can you comment a little bit on that, and can you guide us a little bit?
I think it is tricky because, as you say, it does, you know, you start talking about entanglement and people say, oh, well, you're entangled with other people's minds, and you start talking about invisible interactions between stuff, and people talk about, oh, that explains crystal therapy, and you have to keep a clear blue water, as we say in English, and between what we're doing, I hope, and that kind of stuff. But it is tricky, and you have to be very, very careful. And also, you've got to be a little bit understanding as well, in that often wacky stuff makes people interested, and then they learn something, and then they move from the wacky to the real science. So when I first became interested in quantum biology, it was some of the wacky stuff, the Penrose-Hamerhoff consciousness ideas, but yeah, that made me interested enough to find out enough to say, no, that doesn't work. But there's other interesting stuff there that would work, I think. So yeah, it's rebranding. That's an interesting point, because quantum biology does have a little bit of a reputation. People do slam it and say, no, it's all nonsense. But so far our graduates have all found jobs, so it hasn't been a problem to them. And I still believe that being in that interface between biology and physics is always going to be interesting and important in the years to come. So even if quantum biology, the kind of stuff that we're talking about, goes up in a puff of smoke, the ability to work between biology and physics is going to be important, I'm sure. I see Susanna's hand still up. Do you want to come in here, Susanna?
I have a question when we finish this bit of discussion.
Yeah, okay. Well, why not throw us your question now? Yeah.
All right, sure. Well, you mentioned that maths can be a big stumbling block for biologists learning physics, or physicists trying to explain things to biologists. And I wondered, in your experience at least, what you felt was the biggest stumbling block the other way around for physicists trying to learn about biology, or what was the thing that's sort of most frustrating to biologists that the physicists don't quite get?
Yeah, interesting one. I think biology is easier to physicists than physics is to biologists, and I think that's because of the mathematics. Mostly biology is I mean, I'm going to say something which I should never say. It's a lot of stuff. It's a lot of stuff. You've got to learn about things. But I guess and the hard part of biology, say in evolutionary studies where or phylogenetics, it becomes mathematical. So then it makes it easier for the physicists to get a grip on it. I think maybe the more fluffy, if you like, side of biology, like ecology and stuff like that. But then I still think that's fairly easy for physicists who are interested. I think, to be honest, I think most of the problem is in the other direction. Clarice, you're
I
dying to say something.
respectfully disagree with you.
Okay.
Biology is hard. We are having a hard time catching up. And I think that it's just different ways of approaching a science. And we as physicists are having a hard time to understand metabolic pathways, to understand exactly what happens. So I just want to say that we're suffering here. We're trying our best and we're suffering.
Do you think it is that in biology, it's the vast amount of it? There's a lot of stuff that you have to know that is in biology, in physics, at least I get the principle in something like quantum physics. You kind of build, and it's kind of simple all the way up, and it becomes more complex ideas, but they all sit on top of each other. Biology doesn't have that structure that it all sits on top of each other. It goes off in all sorts of different directions. So, yeah, metabolic pathways and signaling pathways and all sorts of weird things going on in all sorts of directions, and that is hard to get to grips with. And you just need a lot of knowledge, I guess, a lot of knowledge to kind of and be able to integrate it over a lot of years of study. I guess that's probably, yes, back to Susanna's question, that's probably it, because and also I guess as biologists we kind of absorb stuff more holistically, whereas physicists kind of more reductive, and I guess there is that as well. So the holistic stuff is probably harder, but I think as much as anything, because there's a lot of it, and physicists chemists probably are the best people to deal with both, because there's a lot of chemistry as well. I mean, organic chemistry is just as complicated, all of those weird chemical organic synthesis reactions that you have to remember, but yet they also know the quantum mechanics at least a little bit, so they're probably the best people as intermediates. But then they don't like, things like signaling, where it's abstract. You're not looking at a physical reaction. You're just kind of saying, okay, this activates that. And physicists say, well, what do you mean by activate? How does it activate it? Whereas we say, we don't care. It just activates it. And then this gene gets expressed and the cell goes blue or whatever. And those kind of explanations, I think, are frustrating to physicists. We work biologists tend to work at a more holistic, conceptual level, higher up. And we don't feel that we need to dig down all the time. There is this big conflict in biology between the reductionists and the holistic people. And the reductionists are mostly winning, but then areas like systems biology, which is actually my biology base, does try to deal at a higher level as well and bring things together with mathematical equations. Yeah, and actually I'll mention something else, which now I've mentioned systems biology, I'll mention another difficulty, and that is, I'll describe it for systems biology, which is where you make mathematical equations, often say to represent signaling pathways or metabolism. And it's meant that biology is now dividing, as physics did, to a theoretical biology and a practical biology. And there is that. But what happens then, and one of my colleagues who was a modeler, a bioinformaticist modeler, he described the dilemma as so I always call it Kurzig's dilemma because his name's Andrei Kurzig. And he said that when a biologist wants to get a theoretician involved, they want to use the theoretician as their technician, and the theoretician will be unhappy. And when the theoretician is working out their equations and they want to test them, they want to get a biologist involved and use the biologist as their technician, and the biologist will be unhappy. So whichever way you do it, you make someone unhappy. And I think that's part of the problem as well, that we are kind of still in our silo and saying, okay, here's my interesting prediction. Come on, some biologists come and test it, or here's my interesting system. Can't anyone try to model it? And we're asking those questions rather than things integrating from the bottom up. We're kind of doing our own stuff first and then saying, oh, can you make sense of this? So
I think I agree with that. It's a big challenge understanding what's possible for each other to do and just providing that reality check both ways to go, well, that
absolutely.
We can't do that, or that doesn't work because of this.
Yeah, I mean, we can give some problem to a physicist and they say that's far too complicated. Can't deal with that. Okay, and then you have to try to find something that works. Or what we find as biologists frustrating about a lot of the theoretical stuff is give us a prediction, give us an experiment we can do. It's no good just saying this happens this way and then there's this and this and this, and isn't that fantastic? It explains everything. No. And I think this is an interesting cultural difference as well. Biologists are suspicious of theory. We're not really interested in theory unless it can make a prediction that we can test. So when, one that irritates me when I see it is that, what's the theory now that I can't remember, some big phosphate molecules in the brain that someone have made some proposals about them being involved in consciousness. Big phosphate molecules. Yeah, sorry. Polymer molecules. No one's ever seen them. No one's ever seen a polymer molecule. No one's ever detected one. And us biologists say, why are you working on this? No, nobody knows it even exists. So, but because it makes a great model, we can do this, this, and this. The physicists are interested in it. But biologists say, oh, for goodness sake, give us something that's real. So there is that cultural difference as well. We biologists, we have one theory we believe, natural selection, and everything else we're suspicious of.
Thanks.
Any other points, questions? Okay, well, I'll start packing as I'm heading off tomorrow morning, so I hope to see some of you guys in Galveston very soon. And Susanna, you're in London at the moment, I guess, still?
I'm in Bristol, but yeah.
In Bristol, oh yeah, okay, you're in Bristol. But you'll be heading off tomorrow as well?
Early Sunday morning.
Okay, I'm heading off tomorrow. So hopefully I look forward to seeing all of you. And I want to say, the first joke I put up, the one about the two people, you've got to think of what's the punchline of that. So how do you make a man happy and unhappy simultaneously? The quantum superposition of happy and unhappiness. And anyone who comes up with the right caption can, I'll buy him a drink. Okay, don't head off then. So thanks very much, guys. I've got to head off and pack and that kind of stuff. And it's St. Patrick's Day, and I was born in Ireland, so I've got to have a pint of Guinness before I go. So
see you next week,
I'll see you all guys soon.
Bye-bye, John. Thank you.
Bye-bye. Bye.
Wow, for the first time, we finish on time for the first 3 lectures. So I will propose that we take a break for 30 minutes and then reconvene here for a group discussion. So we're the last standing soldiers here, soldiering on. So let's make a break, stop the recording, and see you all again in 30 minutes.